In the interest of time, I think we can get started soon. Can everyone hear me? Okay, great. Okay, so thank you everyone for joining us today. Today it's my pleasure to introduce our departmental seminar speaker, Dr. Lilianna Solnica-Krezel from the Washington University School of Medicine. Dr. Solnica-Krezel is originally from Poland, where she earned her earliest degrees from Warsaw University. She then conducted her graduate work at the University of Wisconsin-Madison. During her postdoctoral work conducted in the lab of Wolfgang Drever, she was a key player in the so-called Boston screen in the mid-1990s, which is really a massive effort to screen zebrafish mutants in early development. She then established her independent lab at Vanderbilt University before moving to WashU in 2010. At WashU, she's currently the Alan A. and Edith L. Wolf Distinguished Professor and the head of departmental biology. Dr. Solnica-Krezel is really a world leader in the study of developmental biology and has primarily used the zebrafish model system to make seminal insights into the mechanisms of gastrulation and early development. And I think more recently, she's pivoted a little bit more towards mammalian systems, which I think we'll hear a little bit more about today. And her contributions to science have been noted by many. In 2019, she served as the president for the Society of Developmental Biology. She's also a AAAS fellow and serves on the editorial board of many journals. And currently, she's the president-elect of the International Zebrafish Society. And with that, I'll turn the floor over to Dr. Solnica-Krezel to share with us her insights about mechanisms of gastrulation from zebrafish to human. Thank you.
Thanks, Nina, for the lovely introduction and invitation. I remember some wonderful visits to Yale University and some lovely meetings in person. It's nice to visit virtually. I hope we will be able to see each other, Nina, in Woods Hole or in other places in not so distant future. Let me share my screen. Okay, it is really my pleasure to tell you about recent studies from our group. As Nina said, something that continues to be our key interest is the process of gastrulation. The process of gastrulation is close to our heart because, as late Louis Wolpert, who recently passed, famously said, it is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. And while in our department, we are studying all of the processes, including aging, in my lab, we are really focused on this short but very influential process in development. Because it is during gastrulation when in human, from a group of cells, which at this stage form this flat sheet of epiblast, all the germ layers, ectoderm, mesoderm, endoderm, and germ cells will be specified. And during process of gastrulation, there will be a specific body plan. We know that this process is conserved in terms of mechanisms of inducing the three germ layers, endoderm, mesoderm, and ectoderm. However, as far as the gastrulation movements, it's a little complicated because, as you can see, zebrafish, frog, chick, mouse, and we know relatively little about human, the shape of the embryos, the shape of the epiblast, so to speak, at the beginning of gastrulation are quite different. Yet, the gymnastics of gastrulation movements, we discover more and more similarities between these model systems. And importantly, at this so-called pharyngula stage, all vertebrate embryos look very similar to one another. Gastrulation has been studied in many model systems, invertebrate and vertebrate. Of course, mouse is a preeminent model system to study mammalian gastrulation. We are very interested how this process happens in humans. And for years, we have and we continue to use zebrafish for obvious reasons. Gastrulation in fish is just about 16 hours. It happens outside of the embryo. The embryo is transparent, both genetically and optically. And we can directly learn from this embryo about mechanisms of the process. In mammals, it's more challenging, particularly in human, where gastrulation starts at 14 days of development and completes about 37 days of development. And at the moment, while significant progress has been made in studying the earlier stages of development with culture conditions allowing take human embryos beyond stages of implantation, all international agreements stop studies of human development at 14 days. This is exactly when gastrulation starts. So what we have been particularly fascinated by is not only these beautiful movements here shown, movements of convergence and extension of mesoderm. You can see these cells moving towards dorsal midline and at the same time extending along the anterior posterior axis. And these movements of convergence and extension reshape similarly all the germ layers. I showed you mesoderm here in red pink. From dorsal to ventral at the beginning of gastrulation, very broad and very short in anterior posterior direction, and very elongated and narrow at the end of gastrulation. Similar movements happen to endoderm, and also neuroectoderm in particular undergoes this convergence and extension. Not only these movements per se are fascinating, but at the time when the cells are moving, they are also deciding what they are going to become. So this coordination of embryo patterning, cell fate specification, and movements is something that is particularly interesting to us. We know that these cell fate specification events are driven in zebrafish, in the frog, but also, as I will tell you later, in mammals, by coordinated signaling of several pathways, including bone morphogenetic protein, Wnt, nodal, and also FGF. Thinking about how cell fate specification, patterning, and movements could be coordinated, the simplest model would posit that morphogen gradients specify cell fates, and once a cell has instruction about its fate, this also determines its movement. But maybe it's a little bit more complicated. Maybe morphogens could work in parallel to specify cell fate and movements at the same time. So these are the type of questions that we are particularly interested to answer. And over the years, we have used forward and reverse genetics to identify genes, but at the same time to map and understand cell movements dynamics during zebrafish gastrulation. And I will focus on mesoderm. Here again, there is this transgenic that highlights predominantly prospective kidney and also axial mesoderm. Dorsal is here, ventral is here. And we are seeing at the same time epibole movements. And now you can see these convergence movements happening, and most of the cells moving to the dorsal midline and extending axis at the same time. But you probably also notice that actually here in this, mostly they move towards vegetal pole. We call it no extension, no convergence zone. In this region, mesodermal cells move as individual cells along quite complex trajectories and relatively slow. However, when we look at this cell population, the morphology of the cells and the dynamics of movements is significantly changing. These cells become elongated along the mediolateral axis and perpendicular to the anterior posterior axis, and the movement is very effective and almost along straight trajectories. In the dorsal midline, as discovered by Ray Keller in frog, we are seeing the famous mediolateral intercalation in the axial mesoderm, and a very interesting combination of a planar and also radial polarized intercalations that are narrowing and extending axes at the same time. The genetic underpinnings of this process in part came from what Mina discussed, mentioned as the Boston screen. We found a number of mutants, particularly in Glypican-4 and Vangel-2, which look almost as normal embryos. They actually have all the parts and tissues that embryos should have. However, they are shorter along the anterior-posterior axis compared to wild type, and everything is broader in mediolateral direction. And you can see it here in the dorsal midline, notochord forming, already quite narrow along the mediolateral axis and elongating along the anterior-posterior axis. And in the mutants, we see the very broad and also unpolarized cells, which is again in wild type. Cells become very much aligned with mediolateral axes, and they do not do so in PCP mutants at this stage of development. So what does PCP pathway do? What type of information does it convey to cells during gastrulation? This pathway is understood best, of course, in Drosophila, where it was discovered, and where it operates in the context of epithelium, where both interactions between components of this pathway, between the cells, but also within the cells, polarize distribution of the components such that some components of this pathway are at the proximal cell edges, and some other components are at the distal cell edges. Therefore, in Drosophila, this pathway is considered as a compass that can polarize cells along the directions of developing tissues. We think that similarly, PCP pathway works during gastrulation. However, during gastrulation, this pathway works in the context of mesenchymal cells. As Brian Siruna initially noticed, and we extended these studies in zebrafish, some components of the pathway, like prickle, for example, become accumulate at the anterior cell edges, while, and the same we saw for Vangel-2, while other components of the pathway, such as the shevold, accumulate at the posterior cell edges. And we have also demonstrated that microtubules, which are organized as in a typical mesenchymal cells via microtubule organizing center, MTOC becomes biased to the posterior side of the cells during gastrulation. So we think that PCP pathway really works as a compass that points in anterior-posterior direction, allowing cells to elongate in mediolateral direction, polarize certain components of the pathway, but also protrusions in mediolateral direction. So if PCP pathway is a compass, how does the compass know where the anterior and posterior is? How does the compass know where the ventral and dorsal is? Some of these questions came also from studies of the original mutants. We know via various genetic experiments that PCP pathway initially operates along the entire dorsoventral axis of the gastrula. So cells initially have this information. However, BMP signaling, which is high ventrally and decreases dorsally, is inhibiting PCP signaling such that in the course of gastrulation, planar cell polarity signaling is only active in dorsolateral regions of convergence and extension movements largely operate, and mediolateral intercalation, mediolateral polarization, while they are inhibited at the highest level of BMP signaling. So this shows the relationship between dorsoventral patterning and the anterior-posterior PCP compass. However, something that continues to elude us is how convergence and extension and PCP pathway is coordinated with anterior-posterior patterning. We know from beautiful studies of Rudi Winkelbauer in the frog that potentially anterior-posterior signaling via activin that forms some type of a gradient is essential for normal convergence and extension. And since activin is not really active in early zebrafish embryo, the nodal pathway would be a great candidate to test for such a relationship. A very talented postdoctoral fellow in the lab, Margot Williams, who now is at Baylor College of Medicine, decided to test this directly. We took advantage of mutants in 1I pinhead, which is zebrafish cryptohomolog and is absolutely essential for nodal signaling in the zebrafish embryo. In maternal zygotic mutants, OEP mutants, you can see in a cross section here that dorsal mesoderm basically fails to form, endoderm also fails to form. So what's left in these embryos on the dorsal side is neuroectoderm. And this neuroectoderm is not really extending properly compared to wild-type embryo. Margot looked at cell behaviors and particularly at this very characteristic mediolateral elongation and alignment, which happens in dorsal neuroectoderm of wild-type embryo. And as you can see, this is completely lost in cryptomutants. So our hypothesis obviously was that PCP signaling is lost in nodal deficient mutants. However, to our surprise, this is not the case. Polarization of prickle is practically normal in these mutants. Moreover, when we have looked at genetic interactions between nodal signaling and VANGL2 or trilobite, we see that polarization of cells is significantly disturbed compared to wild type. However, it's significantly exacerbated if also function of VANGL2 is downregulated. And these studies told us, therefore, that PCP is this major pathway that is regulating anterior-posterior cell polarization. In the absence of PCP, this polarization is lost. However, in nodal signaling mutants, while cell polarity is impaired, they still retain the PCP compass information, leading us to a model that in vivo nodal signaling works largely in parallel to PCP to polarize cells during convergence and extension, and suggesting to us that there must be additional pathways that provide this information to PCP pathway. And I will be happy to talk later, particularly with trainees, what we are doing in zebrafish to look for the eggs. However, now I would like to switch to our initial studies of gastrulation in human. Gastrulation specified germ layers and also shapes germ layers, and we were really looking for a system that would allow us to, system that would be reproducible enough to allow us to do genetic experiments. And at that time, it was very clear that human embryonic stem cells can be differentiated in a variety of cell types. And the question is, can one use human embryonic stem cells also to model gastrulation? And several years ago, Ali Brivanlu and Eric Zigia with Ari Wormflash, who is now at Rice, developed the system, which this is a two-dimensional micropattern gastruloid system, in contrast to standard ES culture, where cells are plated on extracellular matrix and form very irregular colonies. What they have done using microcontact printing, they printed discs of extracellular matrix of specific shape and size. So just as an example, you can see these discs of extracellular matrix on which human embryonic stem cells can be seeded. And when bone morphogenetic protein for about 40 hours, very reproducible and regular patterns emerge of endodermal, mesodermal, and extraembryonic, as well as ectodermal markers. Very talented former student in the lab, Min-Chia Hu, adopted that system. And initially, we have recapitulated all the components, all the characteristics of the system, including activation of SMAD, which is a downstream component of BMP pathway at the edges of the cell, of the disc. And then this really beautiful and very regular staining for numerous extraembryonic, but also endoderm, mesoderm, and ectodermal markers. What I really like about this system, that with the antibody stainings, we can quantify expression levels or the number of cells expressing these markers, as have been really beautifully demonstrated in numerous publications from Wormflash, Ziglia, and Brivanlu group. And also, Kathaliantonakis demonstrated that the system is also operational in the mouse, even though the signaling requirements are somewhat different. So having looked at this carefully, we started to have questions, particularly when we quantified the number of cells expressing markers of ectoderm, mesoderm, endoderm, and extraembryonic tissues. We noticed that they add to more than 100%. We've also noticed that there are some cells that are co-labeling with these markers, suggesting to us that the cellular complexity of gastruloids might be higher. Therefore, we set out to ask several questions. What is cellular complexity of these HES gastruloid? What stage of gastrulation in vivo do the gastruloid platform models? What is the dynamics of germ layer cell-fate specification in this system? And can this platform be used to discover new features of gastrulation in general, and particularly in human? And the experiments that we have done in collaboration with Samantha Morris, associate professor in our department, who is an expert in single-cell RNA sequencing, is to analyze these 2D gastruloids at several stages, at 0 hours, 12, 24, and 48 hours via single-cell sequencing, and try to address the questions that I posed. We have done 2 replicates for every of these stages. And I'll start with discussion of 48-hour culture. You can see 2 replicates. What we have done is non-supervised clustering using SORA system, and annotated these 7 clusters that emerged from that analysis using the canonical germ layer and extraembryonic cell-fate markers. As you can see, we have identified 7 clusters. What was very interesting, even at 48 hours of differentiation, we are still seeing very clear epiblast-like cluster. We observed mesoderm, but it came into flavors, mesoderm 1 and mesoderm 2. I will tell you that this is more similar to epiblast, and we now think it represents primitive streak or mesendodermal precursors. Endoderm and ectoderm. But in addition, this extraembryonic cluster and primordial germ cell-like cells, which was initially very surprising. You can see the expression of these canonical markers of ectoderm, epiblast, mesoderm 1, mesoderm 2, endoderm, primordial germ cells, and extraembryonic. And what was immediately striking to us, that while initially this extraembryonic layer in 2D gastruloids was considered to be trophectoderm, we have seen equally markers of trophectoderm, but also of amnion, which is specific at these stages of development to primate embryos and not mouse embryos. Just to give you an idea about how reproducible this is, not only at the level of antibodies, but of single cell sequencing. We see distribution of cells from 2 replicates very similar for the two experiments. And what is really very satisfying is to look at expression of these markers in replicate 1 in red and replicate not identical, but how similar they are from one replicate to another. I will highlight just a couple of clusters and talk more about typical germ layers later. As I told you, what we were a little bit surprised is to see the formation of primordial germ cells alike. What is characteristic to human primordial germ cells is that they express SOC17, TFAB2C, nano and nanos3. Mouse PGCs do not express SOC17. We can see expression of these, particularly SOC17, TFAB2C, and co-labeling with antibody stainings. And we have done a comparison to previously differentiated primordial germ cells in other labs. And you can see that our gastruloid HPDC cluster is very similar to this differentiation in vitro of PGCs, and also from another set of experiments of directed differentiation of human primordial germ cells. When we compare the module score, which is really comparison of expression of numerous genes specific to PGCs versus the rest of the genes expressed in a specific cell type, you can see that gastruloid PGCs are very similar to monkey PGCs, but quite diverge from mouse PGCs, which express different markers. And in hindsight, maybe this is not so surprising that we are seeing these cells in the gastruloid, because BMP is considered to be a main signal to induce PGCs in the mouse, as shown by Lawson and Hogan many years ago. And BMP4 is also used to differentiate PGCs in culture from human embryonic stem cells. And you can see again this co-labeling with key markers for PGCs in that system. As I mentioned to you, what was interesting also, that in this extra-embryonic cluster, we are seeing expression of CDX2 and KRT7 in at least some cells, markers typical for trophectoderm. However, we are also seeing typical markers of amnion, and our comparison to amnion differentiation and also of human embryo, as published by Chiang et al. Our gastruloid extraembryonic cluster is very similar to amnion and trophectoderm, and we can discuss that in interesting observation later. So what stage of gastrulation this 2D micropattern gastruloid models, and what cell types we can identify that compare to, say, mouse or monkey or even human gastrula. We have used the label transfer function to address this. And in this experiment, what we are using is to use a reference data set, mouse, monkey, and more recently human, and our original gastruloid data, and transfer labels from the reference data to our data in such a way that we are really looking at the closest neighbor, showing the most similar expression of genes, allowing us to determine transfer labels both in terms of stage, but also cell type. When we look at mouse gastrulation, and we have used a beautiful set of data from several stages of single cell sequencing of mouse gastrulation, you can see distribution of human gastroloid cells on this data set. It was striking that the most similar stage was E7, which is beginning mid-gastrulation in the mouse. And when we combine E7 and 7.5, over 70% of gastruloid cells match to these stages. When we just look at gene expression similarity, and correlation is highest again for E7 of mouse development. Looking at cell type identity, you can see the increased complexity of cell types as mouse gastrulation progresses. And when we compare to gastruloids, again, the similarity with E7 and 7.5 is most striking. We have done similar experiments, similar analysis with throughout gastrulation stages. Again, you can see how similar, but also sometimes non-overlapping distribution is between the gastroid and monkey cell types. As far as the stage is concerned, the highest similarity we observed with 16 days post-fertilization in the monkey, which again is early mid-stage of gastrulation, and gene expression similarity is also pointing to 16 dpa. Based on these studies, we predicted that 2D gastroid should be modeling or should be similar to Carnegie stage 7 of human development. And what was very satisfying as we were preparing our manuscript for publication, Tyser et al. From Shankar Srinivas lab posted data of single cell sequencing of one normal human gastrula embryos at Carnegie stage 7. They sequenced predominantly epiblast and its derivatives at these stages. And you can see that our 44-hour time point is most similar to that Carnegie stage 7 embryo. We saw a lot of similarities. For example, if we look at gastroid clusters, there is a very nice correspondence or similarity between epiblast-like and epiblast. When we look at, and also with primitive streak. When we look at mesoderm 1, is very similar to primitive streak in the human gastrula, and mesoderm 2 is very similar to to nascent mesoderm and also emergent mesoderm. In that set, they distinguish many more mesoderm clusters compared to our analysis. Very nice correspondence between endoderm with definitive endoderm of the human gastrula, and also nice correlation for HPGCs. They have not really distinguished a separate cluster. There were just few PGC-like cells that are very similar to PGC-like cells in the gastroid. We had some problems with the extraembryonic cluster of the gastroloid, which shows some similarity to ectoderm and also yolk sac mesoderm in the human embryo. And we are really looking forward to the raw data from that human embryo to be published hopefully soon. So that was very satisfying to see that our experiments in the 2D gastroloid are really pointing to that system modeling the early to mid stage of human gastrulation, because at this stage you can see that primitive streak is already fully extended with the node reaching to the middle of the epiblast disc. So in summary, using single cell sequencing and antibodies stainings, we demonstrated that in this 2D gastroloid system, they are more cell types. Particularly, we see continuous presence of epiblast, 2 types of mesoderm, primordial germ cells, and extraembryonic cell types, which are similar to both trophectoderm and also amnion. So one experiment that I was really excited to do was to ask if the germ layer cells in mammalian system, particularly in human system, would show similar cell sorting behaviors as famously demonstrated by Holtfretter and others. For those developmental biology gurus, you probably remember that what has been done amphibian embryos, but that has been also done by Heisenberg lab for zebrafish embryos, cells of presumptive epidermis and presumptive neural plate, or during gastrulation, similar experiments have been done. These have been dissociated into single cell suspension. and then allowed to aggregate. What was striking was that these cells found each other, so they the same cell type liked each other. They sorted out from the other cell type, and there was, in 3 dimensions, there was also very characteristic arrangement with presumptive epidermal cells being outside and neural cells being inside. To our knowledge, such experiment has not been done with mouse or any other mammalian system, and we thought that having this very regular 2-dimensional gastruloid with all the germ layers, but also extraembryonic cell types, provides an opportunity for genetic property of gastrula stage cells. So Min has dissociated gastruloids and replated them again on these disks of extracellular matrix. What you can see here with antibody stainings is 2 hours after replating, and we see a basically spotty and random pattern, but 12, 24, and 70 hours after replating, we see more localized and segregated, mutually exclusive distribution of gastrula stage cell types. We have seen a lot of movements of these cells, so they are motile, they are proliferative, and they seem to be sorting. We observe this sorting at higher cell density, and it happens faster. And even at higher cell density, we can see already within 24 hours that this exclusion and sorting seems to be happening, which argued that this is not just a matter of proliferation of cells that seeded at certain point in on the ECM. We were, of course, very interested if during the course of reseeding and sorting, these cells retain their identities. So we have done single cell sequencing of 48 hours post-reseeding, and we have identified mostly the same cell types. You can see that the highest correlation is between the original gastroid cell types and reseeded cells, which is consistent with these cells maintaining their identity in post-reseeding. Even though we have seen reduction of epiblast, we have seen reduction of mesoderm 1, suggesting to us that some of these cells actually continue to differentiate along their typical pathways of differentiation. We have quantified this in unbiased manner with using cosine value analysis in collaboration with Mark Anastasio and Shenghua Shi, experts in imaging. And basically, we are just looking at overlap between, say, T and SOX2, and cosine value of 0 corresponds to complete lack of overlap, and cosine value of 1 would represent a complete overlap. We can see that SOX2 and therefore ectoderm and T, we see significant commingling, as you can see here, for example. However, when we look at SOX2 and SOX17, so ectoderm and endoderm, there is very little mixing. We were particularly interested in CDX2, which is extraembryonic cell type, which also sorts very effectively from ectoderm, and so on. We have not done any experiments. However, we now have very rich lists of gene expression that potentially could be explaining this sorting behavior, including cadherins and including efferents. And that's one of the lines of research that we would like to continue with. So what I told you is that the 7 cell types that are present in gastruloid upon dissociating and reseeding can sort and sort with one another. One cell type likes the same cell type and sometimes mixes with another cell type, like, for example, ectoderm and mesoderm, but separates from other cell types. But it's also very satisfying to see that the type of sorting behaviors that have been characterized for frog on zebrafish gastrulae seem to be also operational on these mammalian human gastrula stage cells. And in the last few minutes, I will tell you about something that we have just completed, which is the question, what is the dynamics of the emergence of these cell types during 48 hours of gastroloid differentiation? What are the underlying signaling events that we can at least deduce from gene expression patterns? So you can see here first with antibody staining, when we start at 0 hours just before addition of BMP4, that most cells express these pluripotency markers. And you can see that particularly expression of NANOC and OCT4 are diminishing just barely recognizably at 12 hours in the center. This is more clear at 24 hours and completely absent at 44 hours, but then also present everywhere becomes then limited to the central region corresponding to ectoderm. When we look with brachyury staining, we don't see any staining at 12 hours. We see some staining at 24 hours and then 44 hours. If we think about epithelial mesenchymal transition, which would be marked by downregulation of CDH1, but upregulation of CDH2, interestingly enough, only we see it very clearly at 44 hours, even though we see expression of mesodermal markers already at 24 hours. And then I would like to point out to this very strong GATA3 expression at the edges of the gastroid. It has been recently shown that this might correspond to a commitment of human embryonic stem cells after BMP4 induction to differentiation within very short amount of time. And indeed, within 12 hours, we are seeing this very strong expression of GATA3. So this is with antibody stainings. What else can we now learn from single cell sequencing? What I'm showing you is now integration of all the data sets we have generated. We have sequenced cells just before addition of BMP4 at 12 hours, 24 hours, and 44 hours, and then done clustering and also transferred labels from 44 hours to these cells at earlier stages. What was clear? That at 0 hours, human embryonic stem cells are really different than those treated with BMP4 for 12, 24, or 44 hours. We see some overlap of these 0 hour cells with other regions, but they really form very distinct cluster. And then you can see 12 hours, 24 hours, and 44-hour cells being more distinct. So now looking at cell identities, because these 0 hour cells really seemed to be different, we did not transfer labels from 44 hours. So here they are. Labeling transfer of different cell types showed us that, again, we have the same cell types: epiblast, mesoderm 1, mesoderm 2, endoderm, primordial germ cells, extraembryonic, and sorry, that's ectoderm. Comparing these 0 hour cells to epiblast-like cell types at 12 hours, 24, and 44 hours, it is known that human conventional H1 HESCs are very similar to epiblast. However, we see still that they are somewhat different, and we see that at 12, 24, and 44 hours, we have a lot of overlap between the epi-like cells, telling us that the epi-like cells do not change that much in the course of differentiation of the gastruloid. But you will see that they change in quantity. So here I'm demonstrating the emergence of these different cell types at different stages. At 12 hours, most cells really are similar to epiblast. surprisingly, already at this stage, we are seeing some cells that seem to be ectodermal precursors. We are seeing upregulations of such markers as vimentin and nestin, and within very short amount of time. And I'm happy to discuss that later. At 24 hours, we are seeing again more ectoderm precursors. We are also now seeing clear extraembryonic cell type precursors and mesoderm 1 precursors. However, only by 44 hours, we are seeing all the cell types that I already described to you. And we have done many levels of analysis. This is just looking at germ layer score using numerous marker expression. And again, this really explains, or provides additional support for what I told you, that at 12 hours, it is really epiblast-like mostly, with a little bit of ectoderm precursors appearing. And at 24 hours, less epiblast, ectoderm, mesoderm 1, and extraembryonic cell types emerging. So this told us that this is a very regular process, with cells being distinct from those treated with BMP4. At 12 hours, we are seeing emergence of ectoderm-like precursors. At 24 hours, we are seeing not only ectoderm, but also mesoderm 1 and extraembryonic cell types, full composition of the gastruloid cells at 44 hours. And I hope you notice that the proportion of epiblast cells is gradually increasing in the course of differentiation. Now, can we deduce signaling events from gene expression pattern? I'm summarizing here decades of research by developmental biologists from many model systems. But what is known in frog, fish, xenopus, and mouse, but now I'm showing mouse, that most likely BMP4 from extraembryonic tissues will induce Wnt signaling, and then Wnt signaling will induce nodal signaling, and then different diffusion ability and spreading ability explain what happens here at mid-gastrulation, where there is a lot of BMP4. This is posterior, this is anterior, proximal, distal, BMP4, nodal signaling in red, and Wnt signaling in purple. Arje Wormflash in his lab and with Brivalo and Zigia showed that similar cascade is operating in gastruloids. Everything is triggered by uniform BMP4 application. However, due to receptor distribution at the edge, it is the edge cells that really show strong response to BMP4. BMP4 is inducing Wnt, and Wnt expands. What's not shown here, Wnt is inducing nodal. So this is what we already know, and we are confirming with gene expression this dynamics and providing just a lot of detail about signaling components of these pathways, but also FGF and Hippo pathway that I will tell you about. So starting with 0 hours, we see some expression of nodal components, particularly receptors. Wnt signaling, also a lot of components, but also inhibitors, some FGF signaling component expression, and also Hippo. What I would like to point out that BMP4 seems to be, components are expressed throughout, but particularly strongly at 44 hours. And we see very dynamic expression of Wnt nodal signaling components throughout the differentiation of gastruloid, the same from Wnt and FGF. And I'll just give you a glimpse of what we can learn from this with Wnt pathway. So this is this is integrated 0 to 44 hour gastruloid differentiation, and we can now see distribution and expression of different components of Wnt signaling. Let me just point, for example, DKK1, which seems to be strongly expressed in nascent mesoderm 1 and mesoderm 2, but also in ectoderm. And what is also striking to me is when we look at expression of different ligands. For example, Wind2, very strongly expressed in primordial germ cells but for example, Wind5A, very strongly expressed in mesoderm 2, and Wind6 in extraembryonic cell types. So the data sets we have created provide a wonderful source of formulating hypotheses and then designing experiments, genetic experiments, to test in this model, but also in other models, how these pathways are cooperating with one another in the course of gastruloid differentiation. And we are excited to go in this direction. And just the last few insights, when we compared pseudotime analysis to think about the emergence of different cell types, here we are overlapping gastruloid epiblast, mesoderm 1 and mesoderm 2, with human epiblast, primitive streak, nascent mesoderm, and emergent mesoderm. We see limited overlap with epiblast, but you can see nice overlap with the primitive streak, nascent mesoderm, and emergent mesoderm. What is particularly interesting is endoderm and mesoderm. I told you about mesoderm 1, and with pseudotime analysis, we see two directions, one towards mesoderm 2 and the other towards endoderm. Similarly in human, streak here, one direction goes towards nascent mesoderm, the other an emergent mesoderm, and the other towards endoderm. So this is why, based on this and also gene expression analysis, we think that our mesoderm 1 really corresponds to mesendodermal precursors sitting in the primitive streak-like structure. And can we, the last question I want to address, can we find something new from this very simple system about human gastrulation? And looking carefully at gene expression, the answer is yes, we can gain new insights. Here I'm just showing in mesoderm 1 and 2 expression of many typical mesoderm markers, which is showing us that indeed mesoderm 1 is more similar to primitive streak, and mesoderm 2 to more differentiated type of mesoderm that already underwent internalization and is starting to differentiate. And you can see TBX6 and other markers of lateral plate and paraxial mesoderm. What was, however, very interesting is that we noticed expression of FGF ligands, particularly FGF17 in mesoderm 2. And no or very little expression of FGF8, which is, of course, the main FGF, which is working during mouse gastrulation and during mesoderm formation and mesoderm migration. This pattern of very little FGF8 we observed in gastruloids. We also observed analyzing data of Cynomolgus monkey gastrula. Very little FGF8 in the gastrulating cell populations and very strong expression of FGF17. And it was really satisfying to see this data coming from Tyser et al. When they analyzed this stage of gastrulation. And you can see that at this stage, there is just spotty few cells expressing FGF8, but very strong expression of FGF17 corresponding to this forming mesoderm, and then similarly to our gastruloid. So let me just summarize. What we've discussed is that in this system, we see gradual differentiation and formation of germ layers, reaching this complex set of cell types corresponding to germ layers and also transient cell types such as epiblast and mesoderm 1, and extraembryonic cell types such as primordial germ cells. And what we have also learned that upon dissociation and reseeding, these cells exhibit morphogenetic sorting behaviors similar to those demonstrated for frog and mouse and zebrafish gastrula. And let me thank people who have really pioneered this work in my lab, and that's predominantly one graduate student, but also Mariana Belcheva and Inza Yun. I didn't have time to tell you about their work. The zebrafish work was single-handedly done by Margot Williams, and we could not do this very exciting work without Samantha Morris' help and her lab member, also Sabine Dietmann, a new expert bioinformatician, and our biomedical engineering collaborators on imaging. And let me thank you, NIGMS. They have funded my work on gastrulation practically from year one at Vanderbilt, and I am extremely grateful for their continued support as well, NICHD and Children Discover Institute. And I'll be happy to answer questions. I need to stop sharing because I miss seeing faces.
Thank you so much for that very beautiful talk. Thank you. Yeah, we can start questions now, and maybe perhaps we can start with some of the questions that are already in the chat. Berna, would you like to read your question?
Yes. Thank you so much. This was a fantastic talk. It was really fascinating. I have actually several questions, but I'm just going to prioritize one of them. So I was wondering, this extra-embryonic sulfates you were able to observe, it's very interesting, and like it seems like there's maybe a mixture of amnion and like extra-embryonic together. And I was wondering, with the single-cell sequencing data, were you able to see like some other diverse cell types of trophoblasts, human trophoblasts that we know, or is it mostly cytotrophoblasts? Sorry.
That's a very good question. We spent a lot of time thinking about this cell type. Separate. We asked, do we have a subset of amnion and subset of trophoblasts? But to tell you the truth, these cells look a little confused. So I think that what we are seeing in that gastruloid system might not be cell type that exists in vivo. And as you well know, it has been shown that conventional human embryonic stem cells are not maybe as easy to differentiate into trophoblasts. It seems that it's the naive cells. We have collaborated with Thor Tunisen here, who's shown that naive cells have this potential. I think similarity to amnion is striking. And again, Fu's lab has shown that BMP4 can be used to differentiate amnion from conventional cells. So we are not seeing further differentiation in this time frame, but we have now cultured these gastruloids a little bit longer, and they start doing interesting things. We don't know, we see some morphogenesis, we see some cavities forming. So I'm very curious, what is it? Is it amniotic-like cavity, or is it more along the trophoblast lineage? So stay tuned. I hope to have answer for you in a little bit.
That's very interesting. So you don't start with naive cells then?
No, these are, sorry, I should have emphasized that these are conventional cells. And the reason for it is that it's actually conventional primed human embryonic stem cells seem to be most similar to epiblast. And Tyser et al., by analyzing this single human gastrula, actually confirmed that conventional cells are very close to the human epiblast. And we already know from studies with Thor Tunisen that naive cells do not respond well to BMP4.
Okay, yeah. Interesting, thank you.
Sure.
The next question is from Dominica. Do you want to unmute yourself?
Hi. I put a question in the chat, but there was another one that came up that I found more interesting. And this goes off of what Berna had to ask about naive and primed. You said that the cells you used are primed and that they mimic or are more similar to the epiblast. But I think towards the end of your lecture, you found no overlap with epiblast. I think you said the gastruloids resembled mesendoderm and not epiblast. So I just
no. Sorry. Maybe let me clarify. Actually, the gastruloids at 44 hours have a full composition of epiblast and all the germ layers plus this extraembryonic and human primordial germ cells. When we have done analysis at 12 hours and 24 hours of differentiation, at 12 hours we have seen primarily epiblast with just precursors of ectoderm, and as the time progressed, the epiblast contribution or presence diminished while these other cell types were emerging. Does that answer your question?
Yeah, that makes a lot of sense. I had suspected that too, but I wasn't sure. Thank you.
I had a question that I think you were starting to answer in your answer to Berna, but you said you're maybe trying to push the system a little bit further. I'm wondering how much longer do you think you can kind of culture these gastruloids, and
so
what the implications of that are?
What we have done at the moment is we have overlaid them with ECM, creating more three-dimensional environment, and then that it seems that extraembryonic sulfate just goes wild. They proliferate and they start doing some morphogenesis. We have not analyzed that yet, and we have cultured them for just few more days. And yeah, so we don't know yet.
I have one question. Oh, go ahead.
So I was kind of surprised that the PGC-like cells seem to look most similar to endoderm, because I think Sarani has even in their human SOC17 paper, they've kind of pushed them being more similar to mesoderm. So do you think that's mostly driven by SOC17, or do you think there's something?
No. So sorry, let me then clarify. I highlighted SOC17 as marker typical to human and primate cells, but thinking about broader similarity, that's a very interesting question, Bloma. Nice to see you. Signature actually to epiblast and mesoderm. And as you know better than I do, probably, it has been proposed that in the monkey, primordial germ cells might be arising in amnion, but there might be migrating through the primitive streak. So there might be showing also a mesodermal signature. We actually see both of them, even more the mesodermal signature. Yeah.
Thanks.
Excellent talk. I have one question regarding Wnt, and that is that Wnt1 we have been always considered as a specific marker of the neural crest. Do you see any role for it during gastrulation? Is Wnt1 at all?
So I would have to look. I don't remember Wnt1 being prominently expressed at these stages. So I showed you that these BMP-induced gastruloids have many sulfates. I didn't emphasize that they are many sulfates they don't have. They don't have axial mesoderm, for example, because they are BMP4 driven. And Ali Brivanlow had beautiful paper that if you use Wnt and nodal, then you're inducing axial mesoderm. So we don't see any sulfates that would be similar to neural crest in the system. So I will need to check. However, do we have? I think we have. There is a little bit of Wnt1, but not prominently.
Okay, thank you.
Hi, excellent talk. I had two quick questions for you.